would like to introduce you to immune cytokine IL-12 that, when targeting directly the tumor stroma, was able to actually induce a terminal growth arrest of a highly aggressive rhabdomyosarcoma and induce its differentiation. I have to, okay. So, a major obstacle when treating solid malignancies is the supportive stroma in which these tumor cells reside. And this is mainly composed of, as we have heard, myeloid-derived suppressor cells, M2 macrophages, instructed fibroblasts, and also regulatory T cells. So we wondered, there is the cytokine IL-12 that has the potential to restore M1 macrophage phenotype activity and also to act on Th17 cells and revert them back into Th1 cells. It also activates NK cells and gamma delta T cells and also supports the priming of alpha beta T cells. So we asked if this cytokine applied into the tumor lesion, made this IL-12 possible to flip the stroma into Th1 and induce a measurable immune response. So to answer this question and to direct precisely IL-12 into the tumor lesion, we used it as a fusion protein fused to an antibody that recognizes histone that is located in the nucleus associated with DNA, but becomes visible to the immune system when cells go into decay, which is the case at necrotic areas of the tumor. To prove principle, we challenged mice with human tumor xenografts with the radio-labeled IL-12 construct, and we found increase of 6 to 8-fold compared to unaffected muscle tissue. And because IL-12 ex species specific, we had to evaluate this therapy in humanized NSG mice, which we did by irradiating the NSG mouse, and then we transplanted CD34 positive stem cells, selected stem cells, and we supported the engraftment by the weekly application of IL-7 as a fusion protein, and hematopoiesis is established in these mice after 12 to 16 weeks. And after 12 weeks, mice received their human tumor xenografts, and we decided for the highly aggressive human rhabdomyosarcoma cell line A204. And after 3 weeks, these mice had established solid tumors from about 250 to 300 microliter volume. We then split the cohort in 3 groups. Group 1 received the IL-7 to further support the human hematopoiesis in these cells, in these mice. Group 2 received the NHS IL-12 construct in combination with the IL-7, and Group 3 did not receive the IL-7, but additionally to the construct received IL-2 complex with a monoclonal antibody 602 to allow the specific binding of this IL-2 to the intermediate affinity IL-2 receptor of memory and NK cells, but not of the high affinity receptor of Treg cells. And we treated these mice for only 5 weeks, and then we saw that the tumors of the controls literally exploded, and for reasons of comparison, we had to sacrifice these mice, all cohorts. And we found a significant lower tumor burden and delayed tumor growth in the IL-12 cohorts. However, as we found that the treatment cohorts were performing so well, we decided to keep 4 of each cohort alive and treat these mice until day 100. And in these cohorts, we found in the IL-12/7 cohort 2 individuals that who had growing tumors, and 1 that had a growth-arrested tumor, and 1 animal died, but that was tumor-free. In the IL-12 cohort, we found 3 of 4 animals tumor-free, and 1 animal had a tumor that was also growth arrested. So because, of course, this had a significant impact on survival, as you can see even in the IL-12/7 cohort, where we lost several animals to tumor burden. Because the infiltration of Th1 cells is an independent prognostic positive marker for survival. We checked the tumor infiltrating lymphocytes, and what we saw was that the infiltration was clearly dependent on the presence of IL-12, but we found distinct pattern. While we had in short-term and long-term treatment the same distribution, we had masses of T cells and monocytes and high-class 2 expression indicating an antigen-presenting function of the myeloid compartment. We found also NK cells and many perforin-positive cells. While in the IL-12/IL-7 treatment, we found in the initial and short-term treatment the same pattern as in the IL-12/IL-2, except for the perforin that was not as pronounced. However, after long-term treatment, we had no perforin, we had no T cells, and also a very reduced myeloid compartment. However, we had lots and masses of NK cells. And as expected, we had only very few NK cells and very few expression of class 2, indicating M2 phenotype of the macrophages. So the molecular analysis underlined the distinctiveness of the IL-12 cohorts. We found activating receptors in the IL-12/7 cohort, NKp44 indicating high interferon gamma secretors, 46 that these NK cells are endowed for the killing of regulatory T cells, via uwvideo on the regulatory T cells and NKG2D on the NK cell, and also NKG2E. In the long-term cohort, the survivor had the same expression pattern like the IL-12/IL-2 cohort, which had a distinct expression, namely NKp4030, which is interesting because this endows an NK cell for the crosstalk with dendritic cells and leads to the killing of immature dendritic cells. So this NK cell compartment was composed to support the priming of an alpha-beta T cell response. And in the controls, we only found the basic equipment of an NK cell, but no activating receptors. Th17 cells are composed of iNKT cells, gamma delta T cells, and also NK cells. And as you can see here in the controls in these tumors, we found only a few uwvideo positive cells in these mice, and very amazingly, we didn't find any hint for Th17 cells or the IL-2, IL-12 cohort. But this is in line with that IL-2 inhibits Th17 differentiation and explains why we didn't find CD161 expressed in this cohort despite dense infiltrates, and CD161 is a marker for transracial migration. We were not surprised that we had masses of Th17 cells in the IL-12, IL-7 cohort, and as we know, IL-12 can bias them into Th1 phenotype, which was reflected in the molecular analysis of transcription factors because we found a significant downregulation of ROC, their lineage transcription factor, which led to an inversion of the T-beta-ROC ratio, and we found a significant downregulation of FOXP3 due to IL-2 neglect in the IL-12/2 cohort and IL-12/7 cohort due to the killing by NK cells. We found significantly more CD40 ligand and leading to the inversion of FOXP3 CD40 ligand ratio, so indicating that we had an activated Th1 lymphoid compartment. We found coenzyme K only significantly upregulated in IL-12, IL-2 cohort. The complexity of a T cell compartment is indicative for us or suggests, gives information about how durable an immune response might be, and therefore we checked the beta chains of the variable segments of the beta locus and found from the top to the bottom is one tumor analyzed, and gray square indicates that this variable segment was expressed, very few T cell segments expressed, while the gray squares in the IL-12 cohorts were mostly distributed as a Gaussian distribution, which means we have a complex and polyclonal repertoire. And these black lines indicate that we had the same variable segments with distinct high single peaks of respective length, and in direct sequencing approaches, we found highly homologous CD3 frequencies or identical CD3 sequences in the animals, and we also found convergent CD3 formation, means identical TCRs resulting from rearrangement involving different variable diversity and joining segments. So this indicates that we have a strong and robust T cell compartment with epitope-specific T cells. Back to the tumors, those tumors that had stopped to grow, we were, of course, interested to know what happened to the proliferation markers, and we found that the PCNA and Ki67 indicated in blue color was significantly downregulated in the IL-12 treatment cohorts. Instead, we found an upregulation of multiple suppressor gene P16. And what was very interesting was that HB1 gamma, which is expressed during the cell cycle and involved in regulating the cell cycle, was translocated into the nucleus. And this was very important, as the nuclear localization of HB1 gamma in the absence of a proliferation marker defines the state of cellular senescence, which means cells are terminally growth arrested. There is no chance to re-enter the cell cycle. So we wondered, what is the ultimate purpose of a senescent cell? Because these cells can persist for long times and they metabolize. And we checked toward function, and what we found was amazing. We found linear streak formation randomly distributed in the IL-12, IL-7 short-term treatment. However, in the long-term treatment and in the IL-12, IL-2 cohorts, we found restiform bundles of muscle cells with pronounced cross-striation, while we found only in a cytoplasmic very faint expression in very rare cells in the controls. So the the senescent cells, the terminally growth arrested cells, stopped their growth and started to differentiate according to their original cell fate. Now, the important question is what induced differentiation, and we have, together with others, published that interferon gamma and TNF-alpha in physiologically low concentrations is able to induce cellular senescence in tumor cells. What happened in our case was that, as you can see here, tumor cell, when you use the same tumor cell and that we put into the mice and put in IL-12 on them, nothing would happen. But we exposed them to interferon gamma and TNF-alpha, and we found that they were growth arrested within 5 days, while the unexposed did proliferate. And after 5 days, they also developed this morphology. They became flat and they expressed the beta-galactosidase, which is a marker for senescence. But most importantly, they also expressed P21, which is the cell cycle excitor for the cells, which is in normal myoblasts the factor that draws them back from cycling and allows the differentiation into muscle cells. Moreover, after 5 days, these cells became elongated and multinucleated and formed multinucleated syncytia. So, coming back to the question, may IL-12 flip a detrimental stroma into Th1? Yes, it can, and by restoring M1 macrophage phenotype and also activating the NK cells and reverting Th17 back in Th1 cells. And by the recruitment of T cells, IL-12 was able to create a TNF-alpha interferon gamma dominated environment where the cells started to become senescent and then terminal growth arrested. They differentiated according to their original cell fate. And depending on the cytokine that was combined, they either eliminated the tumors when combined with IL-2 or they lived with the tumors that were growth arrested. So we think that this immune cytokine has a high potential for therapy in solid malignancies. In the end, I would like to thank people from our department, from my group, and from other departments of the Universitätsklinikum, and Robert Handgretinger, who had the idea of the study, and of course, Stephen Gillis, who made this construct. Thank you for your attention.